Hi, and welcome to Pharmacology of the Synapse. I'm Dr. Anna Lee, and in this video, we will learn about synaptic communication. Here is a schematic of a synapse. You can see that inside the synaptic end bulb are tiny little vesicles that are filled with chemicals called neurotransmitters. The synaptic end bulb from one neuron is almost in physical contact with the next neuron, and this site of communication between one neuron and the other is called a synapse. The neurons do not physically touch. Instead, there is a tiny space called the synaptic cleft. These areas surrounding the neurons are filled with extracellular fluid. The communicating neuron's synapse is called the presynaptic terminal, and the receiving neuron's terminal is called the postsynaptic terminal. Chemical neurotransmitters are contained inside vesicles called synaptic vesicles in the presynaptic neuron. Occasionally, there is physical contact between the neurons that directly permits electrical impulse transmission from one neuron to the next. Gap junctions are an example of such transmission, but we won't be discussing this type of communication in these videos. When the presynaptic neuron is activated, an action potential travels down the axon to the synaptic terminals. An action potential is a propagating wave of changing membrane potential down a cell. So the membrane potential of the cell goes from minus 70 millivolts to plus 30 millivolts and back down to minus 70 millivolts. When the action potential reaches the terminal, the depolarization, or change in membrane potential, causes the opening of ion channels, such as voltage-gated calcium channels. Since the calcium concentration is higher outside of the cell compared with inside of the cell, the calcium enters the cell along a concentration gradient. This increase in calcium in the terminal initiates fusion of the synaptic vesicles with the membrane, called exocytosis, and the release of neurotransmitter into the synaptic cleft. The neurotransmitters then diffuse across the synaptic cleft and contact their receptors on the postsynaptic neuron. These receptors can be ligand-gated ion channels or G-protein coupled receptors, and we will go over both types of receptors in this course. For the propagation of action potentials, the neurotransmitters open ion channel receptors on the postsynaptic neuron. This causes an influx of positively charged ions, such as calcium and sodium, into the cell, which then depolarizes the postsynaptic membrane. These depolarization events can be referred to as EPSPs, or excitatory postsynaptic potentials. One EPSP will only increase the membrane potential by a small amount, and usually not enough to reach the threshold required for an action potential. You can have multiple EPSPs that occur one right after another, which would then add up to exceed the threshold for depolarization, resulting in an action potential. This is called temporal summation. On the other hand, you can have simultaneous EPSPs on different parts of the neuron, which will exceed the threshold for depolarization, resulting in an action potential. And this is called spatial summation. In addition to excitation and activation of the subsequent neuron, neurons can also inhibit the activity of the subsequent neuron. For example, instead of neurotransmitters activating and opening positively charged ion channels, such as calcium channels, it would open negatively charged ion channels, such as the chloride channel. This would cause negative chloride ions to enter the cell and would hyperpolarize the neuron, making it more difficult to reach threshold for membrane potential depolarization, and thus reducing the probability that an action potential would be produced by the postsynaptic neuron. Synapses can get stronger or weaker over time, and this is known as synaptic plasticity. This means that the synapses are either able to transmit signals better, or they become worse at transmitting signals. Synaptic plasticity can occur after a short period of time, within 2 to 20 minutes after an event. Synaptic plasticity can alter the strength of neuron-to-neuron communication, and thus alter the strength of neural circuits. and synaptic plasticity is thought to be the basis for learning and memory. Let's take a closer look at neurotransmitters in the synapse. All classical neurotransmitters have a synthesis mechanism that usually involves synthesis enzymes. This is an example of the dopamine synapse from your textbook, which includes synthesis of dopamine from tyrosine to dopa to dopamine. There is a storage mechanism, which usually involves storage of the neurotransmitter within synaptic vesicles. There are release mechanisms that will release the neurotransmitter into the synaptic cleft. And finally, there are termination mechanisms, which can be in the form of reuptake of the neurotransmitter back into the presynaptic neuron, or metabolism of the transmitter to inactive forms. In this course, we will be reviewing these actions of neurotransmitters as well as talking about some atypical actions of non-classical neurotransmitters.